Hey everyone, in this video we are going to take a look at incomplete dominance and codominance, which are two different things, and it can be kind of confusing to tell the difference between the two. So hopefully after this video you will be able to tell the difference between these two phenomena. So we are going to start with an example from snapdragon plants. So if we were to cross red snapdragons with white snapdragons, the F1 generation would be made up of pink snapdragons. Now that's something we haven't seen before, right? Typically, you know, one of the traits disappears in the F1 generation. And if we were to produce the F2 generation by self-crossing or crossing the pink plants to another pink plant from the F1 generation, we would see, let's see, 1/4 red, 2/4 pink, and 1/4 white plants. So let's look at the genotypes. So what's going on here? Now, the red plants have two alleles, are homozygous for this R1 allele. And this is a little different notation or symbolization than we have seen in the past. But we're going to do R superscript 1 for the allele that causes the red phenotype when it's in the homozygous condition. Now the white is going to be homozygous for a different allele. We're going to call this one R2. So R superscript 2. When R2 is in the homozygous condition, we get the white phenotype. Now, when we cross these, all of the offspring have one R1 allele from the red parent, one R2 allele from the white parent, and they are all pink. So the genotype of one, the heterozygous genotype, leads to the pink phenotype. Now in the self-cross here, or again the outcross to a member of the F1, another member of the F1 generation, so we get 1/4 plants that are homozygous R1, 2/4 that are heterozygous, and 1/4 that are homozygous R2. Now these are the red plants, these are the pink plants, these are the white plants. And you could do the Punnett square if you wanted to see how we get this ratio. Now, okay, this is an example of incomplete dominance, and essentially incomplete dominance results when, well, let me talk about what dominance is, right? Let's review what dominance is first. So a dominant allele is, you know, one that causes the heterozygous phenotype and homozygous dominant phenotype. Homozygous dominant phenotype to be identical, right? There's no phenotypic difference between the heterozygous genotype, the phenotype of the heterozygous plants, and the phenotype of the homozygous dominant plants. There's no phenotypic difference between those when an allele is completely dominant. Like, for example, DD tall plants, one of our examples from Mendel, are phenotypically identical to heterozygous tall plants. So the RR sounds like a pirate a little bit there. The big R round plants, round seeds, are phenotypically identical to the heterozygous big R, little r round seeds. However, you know, the R1 red snapdragons are not equal in phenotype to the heterozygous R1, R2 pink plants. So the reason is because R1 is incompletely dominant to R2. R1 is, well, not dominant to R2. R1 is incompletely dominant to R2. So incomplete dominance is when the heterozygous condition is different from the homozygous dominant condition. We have to add one caveat to there. So incomplete dominance let's see, homozygous condition is different from heterozygous condition, and our example is R1 is different from R1, R2, and the condition involves levels of one product. Okay, so what does that mean? Levels of one product. So let's take a look at, let's consider it like this. Okay, so we have a white precursor molecule, and let's say the R gene here is responsible for making encodes the protein, instructions for making the enzyme that turns the white precursor into a red pigment, right? Okay, and let's say the R1 allele equals a functional enzyme, and the R2 allele is non-functional. It could be a null allele. So the enzyme just does not function. Now, let's use this sort of conceptual diagram of what's going on with these different plants. So let's assume that the plants are represented by these little cups of water. 3 cups. And so here we have a genotype. This will be our R1 genotype. This cup here represents our R1 R2 genotype, and this one here, the R2 genotype. Now let's say this is the pigment right here. There's a little, say, a vial of pigment. Now let's say it's the white precursor molecule, and what each R1 allele can do, let's say this is a little robot here, I'll put R1 here. Each of these can take this pigment, and let's say it can take five aliquots or drops of the white pigment, and it can add it. 5. So it can take 5 drops. There's 6 here, but assume I only put 5 here. It can take 5 of those and add it to here, and turns the drops red, adds it to here. And we have 2 of these guys here. Two of these little miniature robots here. They're taking the white pigment, and they can take 5 aliquots or 5 drops of it, turn it red, and add it here. So let's do 10 drops total. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so this will give us this bright red color, right? Because we have 10 drops, and let's say 10 drops of the pigment gives us the red color. Now over here, we've got one little R1 robot, which represents the functional enzyme. Now it can add 5 drops of the pigment. But the R2, let's say something's wrong with this enzyme. There's a mutation which causes this robot to have no arms. It can't add any drops of the red pigment down here. So the color appears pink. Essentially, there's more of the pigment here, giving it the red color, than there is here. So, you know, the water appears less red, it looks pink. And over here, we have these helpless robots who want to, you know, their job is to take the white pigment, turn it red, and add it here. And they can't add any because they're lacking the arms that the robots need, or enzyme needs, to add the red pigment. So the water looks clear, or in this case, white. So now this is a conceptual diagram of incomplete dominance because we're seeing different phenotypes based on the levels of one product, the red pigment, the reddish pigment. If you have enough of that product, the water or flour appears red. Intermediate levels, it appears pink. 0 levels, it looks clear or white. So, okay, so that is the conceptual diagram of incomplete dominance. We're dealing with one product, different levels of one product. And it's going to be important when we get to codominance, which I'll go over now. So one of the best examples for codominance is the ABO blood type system in humans. So this is going to be our example of codominance, or alleles that are codominant with one another. And so, just very briefly, when we think of blood types, and blood types can be very complex, but a simple view of just the ABO blood type system. If this is a red blood cell in humans, well, on the red blood cell there are, let's say, hundreds, I don't know if it's hundreds or thousands, of these molecules called the H substance. And you don't need to memorize the structure here and the individual components of the H substance, which is essentially a glycoprotein. So we've got amino acids linking together these little carbohydrate molecules, and you don't need to know the names. I don't even know, N-acetylglucosamine maybe, and galactose and fucose, I believe. You don't need to remember those, but just know that their H substance is this glycoprotein. There are amino acids in here too on the surface of the red blood cell, and there could be hundreds of these. Now, there is a ABO blood type gene. I don't know the official name. And there are different alleles for this gene. One of them we will symbolize with a I, uppercase I, superscript A. And what this allele does is it adds a sugar molecule called NAGA to the H substance. I'm going to be really embarrassed if this is NAGL. But again, you don't need to know the names specifically, but just know it adds a sugar molecule to the H substance. And IB adds a different, this one would be galactose, to the H substance. So the IA allele, what it will do, if this is the H substance, this is the red blood cell over here. So NAGL, GAL, fuse, it adds NAGA right here. So this gives us the A antigen. Now, IB makes an enzyme that adds a different sugar to this part of the H substance, galactose. And this is the B antigen. So an individual with this genotype will have blood cells. If these lines represent the H substance on the cell membrane of the red blood cell, then it will have the A antigen everywhere we see the H substance. So they would have type A blood. Now, conversely, individuals who are homozygous for the IB alleles would have B antigens on their red blood cells. And you probably know where I'm going next with this. And this would be type B blood. So individuals who are heterozygous, IA, IB, I have to draw the blood cell a little smaller, would have both A and B antigens everywhere we see the H substance. Now, these alleles are codominant with each other because each makes a distinct and detectable product. If we isolated blood cells from these individuals, we could see that, okay, there is an A antigen, which is because of the IA allele, and there is a B antigen because of the IB allele. So that's why they are codominant. The alleles make distinct and detectable products. So they are codominant. So let's review here. Snapdragon, that was our incomplete dominance example. Incomplete dominance example. And blood type, that was our codominance example. Remember the incomplete dominance, deals with when the homozygous conditions are different from the heterozygous conditions, or the homozygous condition here is different from the heterozygous condition. And we're dealing with levels of one product. So here, lots of red gives us the red flower. Intermediate red gives us the pink appearing flower. And no red gives us the white flowers. So different levels of one product, the product being the red pigment. The codominance example, we have homozygous IA, heterozygous IA, IB, and homozygous IB. Now, this one right here, this is one product, the A antigen. This one right here, we get one product from the IB allele. When they're together, we get two products, A antigen and B antigen. The heterozygous condition is different from both of the homozygous conditions, and there are two detectable products. Each allele makes a different product. So that's an example of codominance. Now, one more thing about the ABO blood type system. So it's also a good example of how you can have multiple alleles or more than two alleles involved in determining blood type. So like we said before, each human can have at most two alleles for each gene. And we have IA and IB that are codominant to each other. Now we also have another allele, I, which is recessive to IA and IB. So IA and IB are both dominant, completely dominant to I. So we can list the possible genotypes here. We've already seen these. So phenotype is type A blood. This one is type AB blood. This one is type B blood. Now we can also have IAI, which is type A, IBI, which is type B, and now II, which is type O blood. So that's how we have type O blood. And this I allele is essentially a null allele. IA is functional. It adds one sugar to the H substance. IB is functional. It directs the addition of a different sugar to the H substance. The I allele doesn't direct the addition of any sugar. So it encodes a non-functional enzyme. It doesn't add any sugar to the red blood cell. So if you have type O blood, you've got red blood cells with lots of naked H substance on the cell membrane. Okay, so ABO blood type system, it is a very interesting system and useful example for introductory genetics courses. Okay, so what will the next video be? I think the next video is going to be one of my favorite topics, one of my favorite topics, which is difficult to pronounce, hierarchical dominance. So see you in the next video.